Hi everyone, it's Dr. Hall, and we're going to do lecture 6, which is part 2 of the brain, an introduction to the regions of the brain. So part 1 was accessory structures like the meninges, the CSF, the blood vessels. Now we're going to get started on the actual parts of the brain, and it's really just an introduction because as we learn about other functions of the nervous system, like as we talk about vision or somatosensory or motor function, we're going to get more specific about the regions of the brain involved. So this is your general orientation, and a lot of this hopefully should be review from when you took anatomy and physiology or human biology. So our outline is a few general concepts of how the brain is organized, a brief introduction to cranial nerves because we're going to see those in lab and I want you to have a little bit of orientation to them. And then we're going to talk about the main brain regions, the brainstem, the cerebellum, the diencephalon, and the cerebrum, and some of their subparts. So in 6A, we're going to do general organization and a brief introduction to cranial nerves. I want to bring up some previous concepts that we've already learned. So remember, we have kind of three basic types of functions of the nervous system. We have sensory, which is information coming into the central nervous system, motor signals, which is information or signals or communications being sent out from the central nervous system to the body, and then integration, which is that processing of things within the central nervous system. I also want to remind you of the crossing of tracks when we learned about the formation of the central nervous system in fetal life. Remember, we learned that at some point it twists, so the right side of the brain controls the left side of the body and vice versa. Also remember, of course, those meninges, the three layers, the dura mater, arachnoid mater, and pia mater that surround and protect the brain, and that cerebrospinal fluid that circulates is produced by the ependymal cells in the choroid plexus, circulates through the lateral ventricle, third ventricle, cerebral aqueduct, fourth ventricle, central canal into the subarachnoid space, and then percolates through those arachnoid granulations to end up in the superior sagittal sinus, right? And then it drains into the bloodstream. And then speaking of blood, we have the blood-brain barrier, of course, in our central nervous system, where we have the end feet of the astrocytes helping to help make the wall, the barrier between the bloodstream and the brain tissue a little bit thicker, as well as we have those pericytes. And instead of having loose connections between the cells that make up the walls of the capillaries, we have those tight junctions. Then, of course, remember, if we're in the central nervous system, that the myelin-producing cells that we're going to see are those oligodendrocytes. That's one of those glial cell types that we learned about, where they send multiple different projections out. Remember, I think of it like Elastigirl. So one cell is wrapping different pieces of different axons all around with myelin. So those are some previous concepts that apply, obviously, to the brain. I also want to, you know this, but right, the central nervous system is enclosed by bones, right? So the brain, of course, is in the skull, and that helps protect it, which is excellent. And I just also want to point out that the spinal cord is also enclosed by bones. It travels through this kind of tunnel. If you look at a vertebrae kind of top down, you can see this central canal, this canal in the vertebrae, and that is where the spinal cord travels. So these are very important organs, so they are well protected. And then, of course, they have meninges and CSF too. So most of the time, those bones are really helpful, as I mentioned, protecting from trauma. But as we talked about a little bit in lab, sometimes they can cause trouble. So if you have some bleeding into this space, so this is an example of a subdural hematoma. This is venous bleeding into that space underneath the dura mater, not arterial bleeding, which is a different story. And the problem with subdural hematomas, they don't tend to bleed very fast. They don't tend to bleed into the brain tissue. They're kind of around the brain, but the problem is that blood then starts to squish and push on the brain. So I think you can see, let me get my little pen here. So, here is the blood in this individual, and you can see here is the lateral ventricle that's all squished because the brain is getting squished, and you can see here's this is actually the posterior part of the lateral ventricle, and it's been almost squeezed all the way shut, and in fact the whole brain is a little bit shoved over to this side. So of course you need to alleviate that pressure so that the brain can not be squished. So the treatment is to remove part of the skull in some way so that you can drain that blood out. I mentioned to you that I have a relative who's had this done. This is not actually them. This is a different picture from a different individual. So you can see making some holes in the skull to evacuate the blood, and then they just close the skin over top, and you just have a hole in your skull, which is okay. All right. The other thing I want to point out is that you kind of already take for granted, but it was actually a pretty big deal when scientists first proposed it. So what we learned from Phineas Gage is that the brain is regionalized. So different functions are in different places. It seems obvious to us now, but it was not that way, 100, 200 years ago. So the case of Phineas Gage, obviously the location of his brain injury was places that aren't necessary for keeping you alive, but his personality changed. There's some controversy about that. But so since that time, we've learned a lot about what different parts of the brain do, but there's still a lot of mysteries remain. There's still so much we don't know. The other thing that we see in the central nervous system is gray versus white matter. So hopefully you remember this. So white matter is white because it's covered with myelin. It is actually myelinated axons. That's what you're seeing in this whole region here. This is just all axons. It's just bundles of axons going every which way, carrying signals every which way. Whereas the gray matter, which you can see here, that is gray because there's no myelin, and most of that is neuron cell bodies and some neuroglial cell bodies as well. And then sometimes there are some non-myelinated axons. Remember when we talked about axons and myelin, sometimes if they're really short axons, they're not myelinated because why bother? When we look at the spinal cord, just a little preview, it's interesting to see that the gray matter is on the inner aspect, right, of the spinal cord, whereas in the brain, it is on the outer aspect. So we kind of see that opposite type of structure. The brain is divided into 4 main regions, and so that's kind of when we get into them, we're going to go in that order. So the cerebrum, which is the big wrinkly part, has kind of our higher mental processes. Remember that came from the prosencephalon, that top part of the neural tube. And so that's our more complicated mental processes, often called higher mental processes or fancier things, and it's composed of 4 different lobes. The diencephalon, it also came, I'm going to change my color here to something that might show up. So the diencephalon is in the center here. And the diencephalon also came from the prosencephalon, and it does some really important things like helping us maintain homeostasis and controlling lots of our inner organs. It's also responsible for some things that have to do with mood and memory and attention. So the diencephalon is pretty cool. The cerebellum, located on the posterior aspect here, you might recall that that derived from the rhombencephalon, right, or the lowest part besides the spinal cord of that neural tube, and that's important for coordination and balance. And then the brainstem, which is shown here in green. So the brainstem, interestingly, the midbrain, which came from the mesencephalon, not the rhombencephalon, the midbrain is considered part of the brainstem. I think just because where it's physically located, but its lineage, right, where it came from is different from the other parts of the brainstem, the pons and the medulla oblongata. And the brainstem is involved with life support systems, keeping our organs functioning, and some basic relay tracks and things like that. So we'll learn more about that in this lecture. Excuse you. All right. So before we move too far on and you read things in the textbook, I want to point out that the word nucleus or nuclei gets used more than one way. It gets used for 2 entirely different things. So in the central nervous system, the word nucleus is used to refer to a cluster of neurons that are working together. So it's a group of neurons that are working together to perform usually a specific function, and it's referred to as a nucleus. Now that's very confusing because the word nucleus can also mean that organelle in a cell that houses its genetic material. So depending on which meaning of the word nucleus you're using, it's going to be different. So just be aware of that. When I was first learning about the brain, I found it very confusing. So hopefully now you won't. So in summary, those old concepts are the three main functions of sensory motor and integration: tracks cross, meninges and CSF, blood-brain barrier, oligodendrocytes for myelin. And then the new things that we just talked about are that it's enclosed by bones, it's regionalized, different parts of the brain have different functions. Don't confuse nucleus with nucleus. Gray versus white matter. Gray is neuron cell bodies and unmyelinated axons. White matter is myelinated axons. And then there's four main regions of the brain: the cerebrum, the diencephalon, the cerebellum, and the brainstem. Now I want to do a brief introduction to cranial nerves because you're going to see them on the brains, on the sheep brains that we're using in lab, on the human brains that we're investigating in lab, and you already have seen them. So let's just talk a little bit about those. I like to think of cranial nerves as VIP nerves. So most of the nerves in our body are coming in and out through our spinal cord. So they enter the spinal cord to go up to the brain, or they're coming down through the spinal cord to get out to the body, depending on the direction that the signals are traveling. Cranial nerves are different. They come directly from the brain. So either they're going directly into the brain or they're coming directly out of the brain, or a combination of those. So what we're looking at here is an image from the Hedges textbook, and we're looking at the ventral surface, so kind of the undersurface of the brain. And there are 12 pairs of cranial nerves, so we're completely symmetric, right? We're bilateral organisms. So we have 12, on each side, so 12 pairs. They're given Roman numerals as well as names. So in this particular, I'm going to use the highlighter, I think. Okay, so in this particular image, you can see here and here are the olfactory nerves, and right next to them are the optic nerves. And then it just says cranial nerves. All right, which are all these little brown things that you see down here. So all of those are cranial nerves. So I just wanted to kind of point that out because I think it's a little confusing in the book. Let's talk about those first two briefly. So the olfactory nerve is this one that you see here. It's kind of number one because it's the one that's at the top if you're looking at the ventral surface. And you can see here in yellow all of the other cranial nerves that we will learn about next week. We're going to skip them for now. So olfaction is your sense of smell. So the olfactory nerve is bringing information from your nose. So you can see here on a sagittal view, we've got, let's do the pen. Right, we've got all these little nerve fibers here that are up in your nose that actually then go through little tiny holes in your skull to then come to these guys. And then that's going to take that information back to your brain. So the olfactory nerve, cranial nerve number one, it gets Roman numeral number one, is all about your sense of smell. Cranial nerve number two is the optic nerve. So optic, like optician, like optical, right? It has to do with the eyes. So the optic nerves go to the eyeballs, obviously. So you can see that here. They've kind of put the eyeballs in for you. You can also see those olfactory bulbs or olfactory nerves. And here is a sagittal view, so you can also see how then that nerve is taking that visual information into the brain. And then when we learn about vision, we'll learn about where those pathways go. But the optic nerve is all about vision. It is the nerve from your eyeballs, cranial nerve number two. So that's it for your introduction to the cranial nerves. Pretty short and sweet. And we'll pick up with lecture 6B, where we start talking about the brainstem. Yeah, we're going to talk about the brainstem. See you then.